The COVID-19 pandemic brings us devastating effects in our lifestyles, in our economy, and most importantly, death of million people. These effects are brought upon us by the virulence or severity of the SARS-CoV-2 virus, a virulence that can be explained not only by studying the SARS-CoV-2 virus alone, rather by studying the little scenes behind the interactions between the proteins of SARS-CoV-2 and human cell. This video consists of 4 parts. The introduction, which includes the background of the study, introducing the E proteins of the SARS-CoV-2 and the pulse 1 of the human cell junction, as well as their interactions. In this part, the problems of the study are also going to be introduced. The second part is the methodology. The third part is results and discussion, which includes the findings of the study. And the fourth part is the conclusion. Now let's proceed to the introduction by first discussing the E protein or the envelope protein. So the structure of the coronavirus actually looks like this. It has spike protein, nucleic capsid, and membrane protein, as well as the envelope protein, which is the colored violet in the picture. The E protein also has 2 distinct domains, the N-terminal transmembrane domain and the C-terminal domain. This is shown in this picture. And these 2 distinct domains are involved in mediating host immune responses. The N-terminal transmembrane domain is forming a pore, and also the C-terminal domain is involved in PDZ binding. A PDZ binding in the pulse 1 protein, which is a human cell junction protein. In humans, there are approximately 150 unique proteins, including the pulse 1 protein that encodes 1 or more PDZ domains. These domains are essential in regulating human immune responses. That is why the PDZ binding motif for the PBM at the C-termini of the coronaviruses, just like the SARS-CoV-1 and the SARS-CoV-2, have the ability to hijack the human proteins, potentiating their virulence. However, in normal circumstances, the Crom's C-terminus or the CRB-CT is the one that is actually binding with this PDZ. Now let us discuss the interactions between these 2 proteins. So for example, in an infected lung epithelial cell, meaning the virus is already in the epithelial cell, the replication and transcription of the virus genome produces a high load of E protein. This E proteins then localizes into the ergic site or the endoplasmic Golgi intermediate compartment. And since the E proteins interact with the pulse 1 protein via the PBM at their C-termini, the pulse 1 also then localizes or goes into the ergic site. This led to the epithelial cells junction loosening and leaking. These leaking junctions may likely to promote local viral spread, flow of fluid, as well as the immune cells. This then contribute to the vascular leakage, the diffuse alveolar damage, cytokine storm initiation, as well as the ARDS or the acute respiratory distress syndrome, commonly leading to death in the elderly COVID-19 patients and those with underlying conditions. And this is the reason why studying the protein-to-protein interaction as well as their structure is extremely important, as there is a lack of structural information to define such protein-to-protein interactions, which hinders further understanding the mechanism of the E-mediated virulence. That is why this study aims to describe the structure of the pulse 1 and E protein complex, so that we can define the mechanism of recognition of the pulse 1 domains by the C-terminal PBM of the E protein. In order to maximize the information we can get from our study, We can also try to measure the binding affinity between the pulse 1 and the E protein of the SARS-CoV-2. We can then compare this binding affinity as well as the structure we can get from the pulse 1 CRB-CT complex. Now let us proceed to the methodology of the study. Since our aim is to describe the structure of the pulse 1E complex, we first have to produce the said complex. Thus, our first step is the production of the PSG EC18 complex. You might be wondering why is this PSG EC18 complex instead of Pulse 1E complex. Well, it is just the same. The Pulse 1 has 5 domains, 2 N-terminal named as L27 domains, and 3 C-terminal domains collectively named as PSG. These are specifically the PDZ, the CH3, and the GK. The EC terminal 18 amino acid peptide containing PBM was synthesized, and the PSG, which is our concern, was purified and expressed using the nickel NTA and the size exclusion chromatography. To check the binding affinity between the EC18 and the PSG, we use as the microscale thermophoresis or the MST method. The two proteins were then mixed. The second step was that to determine the structure, single particle cryo-EM was used. The sample was then applied to a glow discharge quantifoil or room grade. The cryo-EM data were collected with the use of the Thermo Fisher Titan Cryos equipped with a Garan K3 camera and a bioquantum energy filter. The fourth step is particle picking, which was done through local picker. And then particle averaging was done using the CryoSpark and Reliant 3. The mop was filtered using the Phoenix, and the model, specifically the 3D model, was refined using mole probability and refinement statistics. Now let us proceed to the third part, which is results and discussions. Now the resulting 3D model is actually this picture. We can see the EC18 being colored violet, this one, and the PSG domains, the 3 PSG domains, the PDZ, the blue one, the GK, the orange one, and the CH3 domain, the green one. Zooming in, we can see that the EC18 is actually interacting with the PDZ domain, the blue one, and the SH3 domain, which is the green one, through the DLL7 motif. See here, this is the D, the L, the another L, and the 7. And this is actually inserted in a hydrophobic socket in between the two said domains. And we can see here that the involved in the hydrophobic socket or the residues in the PDZ domain, we have 5 residues involved. The phenylalanine 318, which is actually this one. Please follow my cursor. And the leucine 321, the leucine 267, the proline 266, and the valine 284. And in the SH3 domain, involved residues are the leucine 403 and the leucine 369. And the result from the MST analysis is that the PSG EC18 affinity, binding affinity, has a low affinity, or it is actually weaker than the normal one, with a concentration of 11.2 micromolar. To understand the structural basis of EC18 pulse 1 binding and its implication with respect to virulence, we compare this complex with the X-ray structure of the PSG CRB-CT complex. And it is showing that 3 domains are actually interacting with the CRB-CT, while only 2 domains are interacting with the EC18, and this is actually shown in the previous slide. Not only the PDZ and the SH3 domains are actually interacting with the EC18. Consequently, in the PSG EC18 complex, the PDZ and the SH3 domains are rotated 38 degrees relative to the GK domain, likely due to the disengagement of the PDZ and SH3 domains from the GK domain. Now let us proceed to the last part, which is the summary and the conclusion. From the reported structure, it shows that the E-protein C-terminal DLLV motif recognizes a hydrophobic pocket formed exclusively by the hydrophobic residues from the PDZ and SH3 domains of the pulse 1. This structural analysis actually provides a support or an supporting explanation for the observation that the viral E protein recruits pulse 1 from the lung epithelial cell junctions. In addition, the structure also provides novel targets for the peptide and small molecule inhibitors that could block the pulse 1 E interactions to reduce the E-mediated virulence, such as a peptide inhibitor with a C-terminal, isoleucine, leucine, or even phenylalanine, so that they can penetrate the packet deeper with the higher affinity. Bearing the results in mind, we can always hope that with the right knowledge and patience, we can end this pandemic together. And that ends my report. This is Junaifa Lawiamir. Thank you once again.